So I'll get started. I'll be talking about how we're using personalized virtual brain models to understand how language develops in childhood. In a unique collaboration with Dr. Darren Cadis at the Hospital for Sick Children and Dr. John Griffiths here at CAMH, I get to combine methods and data to explore this. And so across species, many brain functions are preferentially processed by one hemisphere. It's a principle of brain organization. And in humans, language represents a clear example of, sorry, thank you. It's working now, thank you, yes, thank you. Sorry about that, thank you, okay. So the development of gross language function in the brain is characterized by a transition. And it's unique because it's a very protracted one that spans 2 decades, from engagement of brain regions in both hemispheres in early childhood to engagement of regions mostly lateralized to the left hemisphere by adolescence. And this happens in a window of development, as I said, that spans over 2 decades. From a physiological perspective, this is a transition from an organization that, although is redundant, is more resilient to injury, to an organization that is more physiologically efficient but vulnerable. And despite the importance of lateralization and functional specialization to cognition across species, we still don't understand how a diffuse and highly plastic system transitions into this physiologically efficient and specialized architecture of the adult brain. How does the brain functionally specialize for language? What this lateralized activity that we observe in fMRI and brain mapping in terms of what does this look like in terms of excitation and inhibition? And so studies using magnetoencephalography have provided insights into the neurophysiology underlying language lateralization, showing that it's not just the activation of regions in one or both hemispheres, but a process involving changes in both. Specifically, during an expressive language task, we observed changes in the beta frequency band between 13 and 23 hertz, where we observe a decrease in the left, referred to as event-related desynchrony, and an increase in beta power in the right, referred to as event-related synchrony. And this, based on previous studies in the motor cortex, has been thought to reflect excitation and inhibition concurrently. And recently, we've also shown that these also map their trajectories with lateralization, with the beta ERDs becoming more left lateralized with age and beta ERS becoming more right lateralized with by adulthood. And so the question becomes is, how do we get this coordinated left hemisphere activation and right hemisphere inhibition by adulthood? In order to coordinate left excitation and right inhibition, we know that the brain relies on interhemispheric pathways to transfer and synchronize information across hemispheres, with the corpus callosum being the largest and most significant of those pathways. And it also significantly develops in childhood. But it's still unclear how interhemispheric connections facilitate this. And it stems from an ongoing debate that callosal projections, although they're excitatory, can target either excitatory or inhibitory populations. And so based on the neurophysiology that we observed, we hypothesized that this is supported by a projection from the left pyramidal population that activates inhibitory interneurons in the right, which then reduces the activation from the right pyramidal population and the input back to the left inhibitory, forming this interhemispheric inhibitory loop. And this hypothesis has been supported and proposed by studies in the sensory motor cortex that also demonstrate lateralization of beta ERD and ERS from stimulation to, from studies in stimulating and neural mask models that are also simulating this effect. But also in our own data, using directed connectivity study in our lab shows that the information flows from the regions that demonstrate ERD to the regions that show ERS. And so there's this directionality from left to right. And finally, this is what I like to use as the incriminating evidence. And that is, it recently demonstrated using optogenetic stimulation in the lateralization of mice visuospatial system, that this inhibitory loop, where a pyramidal projection to contralateral inhibitory interneuron supports lateralized activity. But how do we demonstrate this mechanism in the human language system? And this is where we rely and move to computational modeling. And in this case, we're using the Anson Rit, and it's been mentioned by our previous speakers as well. And this allows us to model these projections and see their effects on lateralized activity to actually test our hypotheses through a population of pyramidal, inhibitory, and excitatory interneurons that we then embed to create individualized whole brain models for children performing language tasks. And this is the map that I'll be walking you through, from building the model to validating the model to using it to uncover this mechanism. And we do this for every individual in our cohort for 43 children and adolescents aged 4 to 18 years old, creating 2 models for each, representing the trials in the expressive language task, where in the noun trial, you hear a noun, so car, and you're expected to think of an associated verb, like drive. In the noise trial, you hear speech-shaped noise, so it's still an auditory stimulus, but no response is expected. So 2 models per person for each trial. And we embed the neural mass model within a structural connectome using each individual structural MRI and parcellated brain regions, which is serving as the prior for the long-range connections that we implemented. Specifically, we incorporate long-range connections from the pyramidal population to every other population. So that we will have pyramidal to excitatory, pyramidal to pyramidal, and pyramidal to inhibitory. And although they're all excitatory, then their net effect depends on what population they're targeting. And we simulate the auditory cortices to represent the auditory stimulus in our task. But to compare the MEG activity observed in the task to our simulations, we cast the activity simulated from the population source level through individually derived forward models. So that's another level of personalization that we're adding in our models. And we then compare that to the empirical using Hobbit, which is a whole brain modeling in PyTorch library that it has been developed by John Griffith's lab. And this allows us to cast the whole brain network as a recurrent neural network and train it iteratively, improving the parameters, long range and local dynamics until our simulated activity is minimizing the difference between the empirical and the simulated. Now, to note, we started by fitting our model to just the early part of our task. So this is the part where they have only heard the noun or the noise, not when they are generating the verb. And we wanted to do this because we wanted to start out right at the beginning of the neural cascade to see if, to be able to stimulate it right from the start. And so what we have to check is whether the simulated activity ends up matching the empirical, and this is the data for one subject, and we see that our model fitting works in that the evoked response over time, the temporal evolution of it in both trials, noun and noise match one another, but also the spatial topography of these ERPs. And so this is an initial check to see that our fitting is actually working, but recall that this is not the part that we're interested in terms of where the lateralized beta ERS activity is happening. That happens actually 700 to 1200 milliseconds into the task. And instead of trying to fit our model to the oscillations during that period, what we thought to do instead is, okay, let's take our model and simulate it for a much longer period and see, does it in fact capture that? Will it predict those changes in oscillations? And we do so by comparing we will compute the beta power in the noun and the noise trial models and compute the difference to see if we observe ERD, ERS and their lateralization patterns. And so empirically in the data, what we find is in the older group, the adolescents, the older part of our cohort, we observe an increase in beta power in the right and a decrease in the left that is significantly different, which represents a lateralized beta ERD ERS pattern. Whereas in the young children, there's no significant difference between those patterns. And what we hope to see in the model as well is exactly that. So the model is able to capture these developmental differences in the beta ERD ERS patterns. And noting that, again, this model was not fitted on the changes in oscillation. It was fitted just on the evoked response to the noun versus noise. And so this was very interesting. I myself didn't think this could work, but Jing believed in it. And then we also computed a laterality index for each individual and saw if across the cohort could the model also capture that developmental trajectory of increasing lateralization with age. And it does. So what you see in the black line is the empirical trajectory of our data set, and in the green is what the model predicted. And so what this tells us is that, one, we have a model that is sufficient to investigate what drives beta ERD-ARS laterality. But also interesting to note that it's telling us something about the relationship between early neural responses to an auditory stimulus and later oscillatory changes. And something that this reminded me of is the early work of modeling evoked and induced responses by Carl Friston and Olivier David that hinted at the fact that there is this relation, that they are related, evoked and induced responses, and they're related through these stimulus-induced structural effects, which we will see is what is actually driving the prediction of those in our model as well. And so now that we were able to get a model that is able to produce the phenomenon of interest, we go into the parameter space and see, okay, what is related to laterality? And to answer the question, is it really supported by this interhemispheric inhibition loop? And so what we found was when we hypothesized that the interhemispheric inhibitory loop would drive lateralization, one would think that means this is present in the adults, adolescents, and not the young children, because the adolescents are lateralized, but the young children are not. But what we saw was that it's actually present in both. The interhemispheric inhibitory loop, where greater inhibition from left to right than from right to left, was present even in the young children who are bilateral. And so the question is then what is exactly driving the difference between these two populations? And it's that this interhemispheric inhibitory loop is not sufficient, which is what was been proposed in the previous studies and the mice study as well. And so when we look at the data, we see that interhemispheric inhibition does increase with laterality. And so perhaps it's not about the directionality being left to right, having more than right to left, but maybe overall, on average, the interhemispheric inhibitory strength increases, and that is what drives lateralization. And so to actually test this, what we do is we take the interhemispheric inhibitory circuits of the young children and transplant it through what I call a very virtual interhemispheric inhibitory circuit transplant into the adolescents, hoping that that would make them bilateral. And vice versa, we take the adolescent inhibitory circuit and transplant it into the young children, hoping that it would make it lateralized. And so what I'm going to show you is the pre and post results of this virtual transplant. As you know, pre, we see the significant difference in adolescents. After giving them the young children circuits, we see that that was enough to diminish the laterality in the adolescent circuits. And likewise, in the young children, they start out being bilateral, but then with the adolescent circuit, that didn't work. So it only worked in the young children, but not in it only worked in the adolescents, but not in the young children. In the young children, giving them the strength of the adolescent interhemispheric inhibition was not sufficient to make it lateralized. And so what else is going on? We look at the local dynamics in our system as well, and we see that actually local inhibition reduces with age. And this is interesting because in many sensory systems, high local inhibition is what prevents premature specialization and lateralization. And so perhaps this already high local inhibition is preventing us from changing the laterality of the young children's system. And so to change another hypothesis is that what I just stated. And so to test this, we take the young children, we reduce their local inhibition, and in the adolescents, we increase their local inhibition and see the effects of just that on laterality. And we see in young children, that was sufficient to induce lateralization. And in the adolescents, that was sufficient to diminish it. And so what does this all mean? I tried for a long time to come up with an analogy for these findings until one day my mentor said, oh, so the fossil was there all along and development is just brushing it off, and said it so elegantly. And yes, and it actually also looked like it was there all along. We just didn't see it. The fossil here is that intrahemispheric inhibition, specifically greater left to right, is present early on in childhood. This could be developed prenatally or a genetic coded thing, but it doesn't have an effect on the functional lateralization of these children's networks because of the high local inhibition earlier on in development. And that is when this local inhibition reduces by adolescent, that they can, that their networks actually lateralize. And so with that, I guess the mechanism that we're proposing is local inhibition is what allows the intrahemispheric inhibition to drive laterality with age. And going back to this idea that lateralization is a principle of brain organization, and these mechanisms that seem to be appearing in many systems, and this could be a potential to find this unifying mechanism for how the brain organizes function. And thank you very much. I want to thank everyone who has been with me along on this journey, and for you all for listening. Thank you so much.